year's Nobel Prize in Physiology and Medicine was awarded to Dr. Yoshinori Ohsumi for his work in the area of autophagy. Dr. Ohsumi is the 25th Japanese scientist to be awarded the Nobel Prize. He was born in Fukuoka, Japan in 1945, spent some time in Rockefeller University before he came back to the University of Tokyo as an assistant professor, which is where the work that led to this Nobel Prize was done, and is currently a professor at the Tokyo Institute of Technology. Autophagy is the process of self-renewal. The discovery of this mechanism and the steps that are involved in this is what won Dr. Ohsumi this prize. The term autophagy comes from the Greek meaning self-eating. It's an evolutionarily conserved process by which cytoplasmic cargo is sequestered inside a double membrane vesicle and delivered to the lysosome for degradation. Autophagy substrates include organelles such as the mitochondria, aggregates of proteins, and in diseases like neurodegeneration and also various pathogens. The discovery of autophagy is linked to the discovery of the lysosome itself, which was made by Christian Didoué in 1950 using techniques that Albert Claude developed. Albert Claude, Christian Didoué and Georges Palade won the 1974 Nobel Prize in Physiology and Medicine for the discovery of the lysosome. Christian Didoué during these studies also found portions of the cytoplasm to be sequestered into membranous structures during normal kidney development. Recognizing that these structures had the capacity to digest parts of the intracellular content, he coined the term autophagy in 1963. In the early 1990s, Dr. Ohsumi decided to study autophagy, but in a new system which is the budding yeast Saccharomyces cerevisiae. The yeast vacuole, he found, is the functional equivalent of the mammalian lysosome. He found that autophagic bodies accumulated in the vacuole when the engineered yeast were grown in nutrient deficient media, which allowed him to now develop a screen for mutations that would disrupt this process of autophagy. This led to the discovery of the first gene that he called autophagy 1, APG1 for short, and also the discovery of 15 other genes, which was published in a study later. These genes were named originally as APG1 to 15 and are now called ATG1 to 15 as well. The major steps in autophagy involve the development of this phagophore, which is a double membrane structure that expands and engulfs the material that needs to be broken down. The completion of the vesicle then leads to its fusion with hydrolytic enzymes and the lysosome, which then breaks down the material that's inside the cell and makes it available back to the cell itself. This leads to the formation of the autophagosome that is shown in this particular slide. There are different types of autophagy: macroautophagy, chaperone-mediated autophagy, microautophagy, which then brings us to the all-important question of what autophagy does for cells. It plays an important role in maintaining a balance between homeostasis and stress adaptation. It provides nutrients during catabolism. It plays a role in generation of ATP in stressed and starved conditions. It signals for heterophagic removal of apoptotic cells, for degradation of misfolded proteins, removal of surplus or damaged organelles, and also contributes to genomic stability in tumor suppression. We thus find autophagy could act as a safety net in certain circumstances. It could also play a role in processes like cellular development. Many aspects of development are involved in the process of autophagy. It also has a role in immunity, playing a role in adaptive, innate and other kind of immune processes. Autophagy is also a mechanism by which many bacteria and viruses make their entry into cells, and so autophagy also plays a role in bacterial killing and is regulated in that manner. When it comes to diseases, it's now been known that autophagy is deregulated in number of pathologies. You have things like diabetes, obesity, where autophagy is altered. We also find that autophagy has a very strong link now to neurodegeneration. Many molecules that are involved in the process of neurodegeneration are all contributing to regulatory steps in the process of autophagy. It also has pro and anti-cancer properties, which plays an important role in tumorigenesis of cancers as well. As a result of all this, autophagy has also become a very important drug candidate. So many molecules that are shown in this slide have been developed to target different steps in the autophagic process. I'll end by leaving you with this link to a set of recommended reading material on autophagy itself, which will tell you a little bit more about this process and its significance in today's science.